So good morning, everybody. Welcome to the workshop. I'm Professor Sumak Dara in the Department of Statistics, and I have my colleague, Professor Vivek Roy, also in the Department of Statistics. And we are going to introduce today's workshop. So can you see my screen? It should show the slide for SVENETICS.
Yeah, we can see it.
Yes.
Okay, perfect.
Yes.
Thank you. This workshop is supported by the Agricultural Genomes to Phenomes Initiative, project award number 2023 704124187 from USDA and IFA. But any opinions, findings, or conclusions or recommendations expressed in this publication or in this workshop are those of the authors and should not be constituted to represent any official USDA or US government determination or policy. First, I would like to acknowledge Eddie and Shooting for their help organizing this workshop. And today we have two presenters leading this workshop, Deboshi Chakraborty and Srijit Roy. Both are PhD students in the Department of Statistics, and they have been working on high-dimensional statistical methods. One of those problems are GWAS and TWAS for high-dimensional genetic data. And Deboshi will be leading the workshop, but we also have Srijit and myself, Professor Roy, and another RA Tiruni to help you with. Later on, we will have hands-on sessions, so we will be happy to help during that time. So broadly, today's agenda would be divided into three parts. First, Deboshi will be presenting on SVENETICS and demonstrate the software. After that, participants are welcome to stay and reproduce the results from the workshop. We will provide the URL to the data once we start that part. And later, participants are welcome to try SVENETICS on their own data, and we will be happy to help through the process. There will be several steps, and we'll be happy to help through the process. So with that, and the references. So the paper for SVENETICS is still not published. We will email the citation to SVENETICS and also any updates to the package to the email list. But for now, you are welcome to use the first two references if you would like to cite SVENETICS in your work until the SVENETICS paper comes out. With that, I would stop and let Deboshi take over.
Okay. Hello, everyone. Shumakta, can you confirm if I'm audible properly?
Yes, I can hear you.
Okay. So I'm really happy to lead and present this session. I will try to make it as familiar as possible. So, and in the meantime, if you feel I'm going too fast or if you have any doubts, please feel free to stop me and ask any questions you have. So let me share my screen and start away.
And on that note, actually, we will be monitoring the chat. If you would like to send questions over chat, that would be okay. We'll be monitoring the chat.
Okay, so can anyone see my screen?
Yeah, you can see your SVENETICS slides. Yes.
Okay, fine. So I will start the slideshow and hope it's visible.
Yes.
So, yeah, as mentioned, today we will introduce a new tool to perform genome-wide association studies where there are multiple traits present. We call this tool SVENETICS, and I will briefly go through what SVENETICS is, a brief overview, and the main focus of this session will be how to use it. So what kind of data we might encounter, how to prepare that to put into our pipeline, etc. And I'm happy to take any questions in the meantime. we will sort things out and work together. So let's start. So first of all, let's go through what we are going to do today. So first, we are going to introduce that what problem are we interested in, or what question we are trying to answer. Next, we will see that what kind of data we might have that we want to analyze using our tool. And then we will see how to use the SVENETICS app that we have built to answer our question. These are the main three goals of this workshop. So let's get started on the first one. So genome-wide association studies, which many of you might be knowing better than me, involve discovering single nucleotide polymorphisms that are associated with the phenotypic variation in different traits of interest. So we will slowly get into examples and make ourselves more familiar. So this is basically the structure. We have some particular trait of interest, which say for the time being might be the height of a plant or the flowering time. And we have a lot of single nucleotide polymorphisms. The number of SNPs might be thousands or typically millions. So what's our goal? Through GWAS, we want to know which of these SNPs, which are usually very few compared to the total number of SNPs, are responsible for explaining the variability, the phenotypic variability present in this trait. That's what we want to do. That's our problem from a very broader perspective. Next, we will get into a little bit more detail. Say, let us start with an example. So this is a paper by Leibov and others, which was published in Nature Communications in 2015, that genetic control of morphometric diversity in the maize shoot apical meristem. We will call this shoot apical meristem in short as SAM. So they provide a very nice example of a data and try to analyze that with GWAS. So basically the SAM volume that they have observed that it is very much associated with different agronomically important traits of the maize plant, such as flowering time, stem size, and many other things. So they were very much interested in this particular trait, the SAM volume. So the SAM volume in their example act as this trait. And they also have 1.2 million SNPs associated with that. Now, going back to our setup, what question do they want to answer? That which of these SNPs are important? Important in sense of what? Important in explaining the phenotypic variability present in the SAM volume of different genotypes. So this is one example of what kind of problems we might be interested in. Okay, so next let us see what we have. So to perform GWAS, of course, a lot of research has been done till date. And let us see that what are some existing methods through which we can perform GWAS. So two of the methods which we primarily have studied especially to see that whether our method works better or worse than that is this farm CPU by Liu et al. And this Blink by Huang et al. Both of them have R packages available in GitHub, so they are pretty easy to implement. So it was pretty easy to implement for us also, the comparative studies. And there are other methods also like GLM, MLM. These are mainly based on mixed linear models. And these class of methods as EMA, Emacs. Emacs is basically EMA expedited. This GEMA is also a variant of EMA and things. So these are some broad overviews of what the literature is already there. But we, in our comparative study and also in our paper, have focused mainly on these two, FarmSoup and Blink. to do our comparisons and see how their approach differs from their outputs differ from our outputs, etc. Okay, so this FarmCP and Blink will also come up next. So we have this new method, which is called SVENETICS, and we would like to introduce you to it. So today, this workshop, the main goal will be to familiarize all of us with how to use it, how to prepare our data, how to use it, and how to interpret the outputs. So let us see, we will give a brief overview of the method as well. So we have this method, which I will not, as of now, go into very much technical details. Rather, I would show you that there should be some reason why you might want to use this method, right? So in some sense, our method should work better or as good as the existing methods. So we performed some simulation experiments where we know the truth. As in, we know which will be the important SNPs so that we can judge or we can evaluate the performance of the existing methods and our methods. And then we can compare them. So the setups of the simulation experiments that we consider are very much motivated by this paper where they introduce Blink. And to judge the performance, we will use two metrics. So what are the two metrics? One metric is called false discovery rate, which is defined as the proportion of discovered or detected SNPs, or we might call it hits as well. I will use these terms interchangeably, which are not truly important. So as an example, we might think that suppose we have a method, and since this is a simulation setup, we know which the true SNPs are. Because suppose we have this X matrix or the matrix of SNPs, and we have artificially created the trait. So we exactly know which the important SNPs are. Okay, now suppose we have a method. That method produces some hits, say 20 hits, or say 10 hits. Now, out of those 10 hits, we see that 7 of those hits are actually the truly important SNPs, but 3 of those are not. So in that case, the false discovery rate will be 3 by 10. That's how false discovery rate works. So it should be pretty intuitive that we want our false discovery rate to be lower. Since among the detected SNPs, how many are not important? So we want that to be lower. And in contrast, we have another metric which is called true positive rate. Now suppose, on the contrary, we have
I cannot.
Yeah, I think he froze.
Yeah. Okay, maybe.
Let me call him.
we see that in those hits, we find 5 of the true SNPs because we know what the truth is. In that case, our true positive rate will be 5 by 10, that is 0.5.
You got cut off for a bit, and I think, but I think it's fine in the sense. Does anybody have any question with regards to the false discovery rate or the true positive rate?
Yeah, please go ahead.
You can either post on chat or just unmute.
I think it's better to repeat, please, the true positive rate.
Okay, sure. I'll repeat it. Go ahead and repeat. So, as I explained, that false discovery rate, we want it to be lower. On the contrary, we have true positive rate, which is defined as the proportion of truly important SNPs that are detected. So for example, let's say we have 10 SNPs that are truly important because we know what the truth is since it is simulated data. Now, among the hits that are produced, we find 7 of those true ones. Then our true positive rate in that particular case will be 7 by 10. So among my, say I have some number of true SNPs, how many of them can I detect? That's the true positive rate. Does that make sense?
Yes.
Okay. So in this case also, it should be pretty intuitive that we should want our true positive rate to be as high as possible. Like if we are making more hits, we want them that they better be the true ones and not the false ones. So we will, of course, there is a trade-off between these two, and we'll see what happens when we compare the 3 methods. So as of now, we should just keep in mind that we want our false discovery rate to be lower and our true positive rate to be higher. So let's now dive into the comparative plot. And I'll explain the plot in detail. If anyone have any questions, please stop me and ask. So this is the plot we have. So on the x-axis, we have the false discovery rate. And on the y-axis, we have our true positive rate. And the green line, this indicates the performance of farm CPU. The orange line, that indicates the performance of Blink. And the blue line, that indicates the performance of our SVENETICS. And all these are in percentages. So I defined them in proportions, but I've just multiplied them by 100, so the figures look better. And they are expressed in percentages right now. Now let us focus on a particular portion of this map, or this diagram. So suppose at a particular false discovery rate at this point on the x-axis, all 3 methods have a false discovery rate of 10%. Sorry, here. So now, given if we fix the false discovery rate at 10%, that is, we are making 10% of our discoveries to be not important, we should watch out that we better are getting some of those true SNPs. So here we can see that SVENETICS at this particular FDR is providing a much higher true positive rate compared to the rest of the methods. Similarly, if we go to any point on the x-axis, say at 20% FDR, we can see at that particular FDR also, the true positive rate of SVENETICS is much higher than that of Blink or farm CPU. And as we go through the x-axis, we can see the same picture. So the takeaway from this plot is that at any given FDR, SVENETICS can achieve much higher true positive rate, which in essence is a sense of power of a method to detect true variables or true SNPs. So SVENETICS can achieve much higher true positive rate and at any given FDR compared to Blink or farm CPU. We have seen this from different simulation studies, different kinds of simulation studies. I'm presenting just one of those diagrams. So is everyone good with me as this diagram? If anyone has any questions on this plot, please feel free to ask. Okay, I will check the chat once.
We are monitoring the chat. There are no
Okay.
questions.
Okay.
You can go.
Thank you. Okay, then let us proceed. So this might motivate you that there is some reason to use SVENETICS as well. So let us proceed. So we want to give some disclaimers before starting to use the app. that generally we have seen that the data we get is stored in a numeric format. So, but our app requires that the data containing the SNPs, the metrics containing the SNPs, requires to be provided in a sparse matrix format so that we can make computations faster. But how to convert your numeric matrix to sparse matrix format, we also have built a separate app to perform that. So if you just upload your numeric format, I will also demonstrate that one. I will demonstrate that app as well. So that will directly convert it to the format that our GWAS pipeline needs. And also the genotypes names between the metrics containing the SNPs and the traits should match ideally. Since we are running an automated pipeline, if we see there are mismatches, we'll drop them. And the third point is very important. We will do not do any kind of filtering based on linkage disequilibrium in our pipeline. So if you want to do it, it is good to do before you start using this app. And one nice option to do is this tool called Tassel. So yeah, that's some points we want you to know. They are also in the landing page of our Shiny app, and we will keep updating it if something comes in our mind or as we receive feedbacks. Okay. So as traits, we will typically have the genotypic blues or blobs, which are essentially the best linear unbiased estimators in case of fixed effects and best linear unbiased predictors in case of random effects. That should be stored in a CSV file where the first column of the file should be genotype names. I will shortly show you the example of such a file so that it's clearly understandable. And what other things do we have other than the traits? We should have the data on the SNPs, which should be stored like this in an RDS file. And the row names of that RDS file should be the genotype names. But this storage, we will take care of it. You just have to have your numeric format, which again, we will show how that should look like ideally. And our tool will convert it into this desired format. So this is a typical example how the trait file can look like. So this first column, you should have the names of the species. Yeah, question?
I
Yes,
think that's an accidental unmute, so
okay.
you can go ahead.
Okay, no problem. So, okay, this is a typical example of a file containing the traits. So the first column contains the genotype names, and next column onwards, we can have trait 1, trait 2. This do not have to be named as trait 1, trait 2. You can name whatever you want. Like the output files will be saved with the same names. And I want to emphasize on a point here that you see that we might have some values missing. So what we will do is we basically carry out the analysis for all these traits separately. So when we are carrying the analysis for trait 2, since these values are missing, we'll drop these rows. But don't need to worry, we will not drop these entire rows because since we are carrying the analysis separately, so when we are analyzing trait 1 and finding the hits, these rows will be preserved, these 3 observations. These missing values will only be dropped for which particular trait the missing values are there. Next, the numeric format we ideally expect to have is something like this. The first columns should contain the names of the species, and the next column onwards will have these values of SNPs like 0, 1, or 2. We have worked with both kinds of matrices where they're encoded as 0, 1, or 2, and some matrices there are only 0 or 1. So again, we can take care of both. You don't need to worry about that at all. We can handle both 0, 1 and 0, 1, 2 matrices. So, and this conversion to the sparse matrix format, this is just an overview of how a sparse matrix looks like. This will be done by our app. And this, we basically do it because this is much more memory efficient and makes the computation faster. Okay. So now we will go and dive into our hands-on session. And I already have these 4 or 5 lines of codes that are ready for you. So this is how we typically install packages in Bravo and packages in R. So the name of our package is Bravo. And this is very important that we require the version 4.1.0. If you have some older version, you might see that there is no such function like this dense to sparse converter or the SVENETICS because it's only in our latest version. So dense to sparse converter launches the app which helps us convert the numeric format to the sparse format. And SVENETICS is our main app which has multiple steps but helps us run the multi-thread GWAS. So now we will dive into R and get some practical essence of how to use the app. Okay, so now I will exit the screen. And first of all, I want to tell you that we have all the data and codes ready. So I will show you how to navigate to the URL, and I will also post the URL of the folder in the chat of the Zoom so that you can access it easily. So if you just navigate to Professor Shomak Datto's website.
Okay. I've already posted the link on
Okay.
the chat. So.
Okay, that's good. So if you open the link, it will look something like this. It's stored in a box folder, accessible to anyone.
And the slides are also given. So
Yeah,
if you
the
would
slides
like
are
to...
also given in PPT format, PDF format, both. And the files we will need for today are all given here. So yeah, we are all good in terms of what files do we need.
So generally, which ones do they need?
They will need this Excel, the CSV file, which says
The trade workshop, yeah.
workshop.
And
They will need the SVENETICS trained this file.
but they can also get it from SNP workshop mat, right?
Yes, that will take time. That's why I have already posted it, because that will take a lot of time.
Sure.
And they will need the SNP workshop mat. That's it. And if someone wants to download this already written R file, you don't have to write any code. The code is the few lines of code, the 4 or 5 lines of code you need is already written there. You can just run it. So I will just show because I already have it downloaded. So the file folder is called AG2PI workshop. If I open it, I will open this AG2PI workshop codes, which has all the code needed. Okay, I hope everyone can see my RStudio.
Yeah.
Okay. So the step to install this package is install.packages Bravo. I already have it installed, so I'll skip this line as of now. Then we'll load the package. And again, very important, we'll check that if we have the updated version or not. So yeah, you can see I have the
Devarsha, do you want to give them 1 or 2 minutes if they're doing it parallelly?
Of course, I can stop for 2 minutes and let you do this. So yeah, it might take a minute or two to install, and once everyone is done, we can resume after that.
So there was a question on chat whether I think lower FDR or higher FDR implies higher TPR. And it's not necessarily. The numerator and the denominators are different. So both of these are ratios, as you know, as Devusi explained. Both of these quantities are some ratios, but the quantities which appear in the denominators and the numerators are different. So yeah, not necessarily one higher values for one implies higher values for the other.
Thank you, Dr. Roy, for answering that question.
In fact, if you get a few false hits, if you get more false hits, then your true positive can still go down, can it? Your false discovery will go up, but your true positive should remain constant.
Yeah.
So it may not go up.
Yeah.
Devusi, I think you can carry on now.
Yeah.
Okay. If anyone is not done yet, you can just post in the chat or unmute and stop me. Otherwise, I will carry on. So first of all, we will see that how to convert a numeric format matrix to the format we want, that is a sparse matrix. So this line of code launches the app to do the conversion. So I will just run this line of code. And I will always recommend that you use this open in browser option. For shorter tasks, it might not affect a lot, but some tasks in this app will take a while. And suppose your screen turns off, the browser window always remains there. The small app pop-up might not. So this is always recommended to use the open in browser option. So now we need to upload our genotype file, which this is just an example that .txt.gzipped. It can be .mat or any other kind of format. So I'll go to browse. In my computer, I have this SNP workshop 2026.mat, but the auto recognize is not showing it because there I have .txt.gz. So I will just make it that it shows all the files, and it generally handles any kind of numeric format. So I'll upload this one. Everyone should have this in their box folder. So I'll open it. And then the number of genotypes is basically, we just need a maximum number of genotypes to read. So for this example, I think we approximately have 368 genotypes. So any upper bound like 500 or 1000, anything greater than 368 should work. It's just a check for us. And the output file name, you can, like, anyone can edit this as they want. By default, we store it as sparsemetrics.rds by this name. And definitely you can also choose like where to save the output. Like as of now, we have these three options. Like, of course, we have working directory, but sometimes some individuals might want to navigate the saved thing easily. So downloads and desktop are pretty convenient options. So I will choose desktop as of now. And this is one more tool, that is the separator. So these numeric format matrices are typically separated by tab, comma, space, or semicolon, something like that. So suppose we start with the tab and I click this convert to sparse. It throws me an error. So why does this throw me an error? Because the file we have currently is separated by spaces and not tabs. Since I provided this wrong, so it can't really identify the SNP columns. So I will choose space And in the next version of our app, we will do this thing like an auto detect kind of thing, so that users don't have to worry about it that much. So now, since I know that it's separated by, it's a space delimited file, now I will click the convert to sparse, started the converting, and it's pretty fast. So it will also show you exactly where this is saved. So it shows me that it is saved in my desktop with the file name sparsemetrics.rds. Okay, so I'll close this one and go back to my desktop and see, I can see the sparsemetrics.rds. This is what we will need. And in this AG2PI workshop, I can keep this one. And just for the case this app doesn't work for someone as of now, we also have a file x_workshop_2026.rds. These two are same. You can use that one as well. This was just to demonstrate how the conversion works. Okay. Now I will go back to my RStudio, and then I will launch our main app, which will help us perform the GWAS. Okay. Again, it's highly recommended that we open it in the browser so that if the screen turns off, it remains. The first landing page basically describes what I briefed to you about the app, how to use this and everything in the presentation. So now we will navigate through the app and see how to use it. This app has two steps. First one is the train step. So this is very interesting. So just a brief discussion that our method has some particular parameters, which in this particular pipeline, we somehow tune those parameters to get the optimum results. And it turns out that the choice of those parameters depends only on the metrics containing the SNPs and not the traits. So we will just provide the metrics of the SNPs, the sparse metrics we already have now. If someone already has that, they have. If someone has a numeric format, they have it converted. We will provide that, and it will calculate the optimum values of the parameters and return us an object that we will need in the next step. That's it. That's the purpose of this training phase. And this training phase, if you note, this is a one-time job, because like SNPs or the SNP metrics for a particular panel doesn't get updated very frequently. So once you have this trained, we can use this to analyze as many traits as we want. So this is a one-time job. This will take time, but this is just a one-time job we do. And once we have this object stored, we can analyze as many traits as we want, which corresponds to the same diversity panel or the same SNP matrix. So I'll go ahead and upload my sparse matrix, which we just stored. And next we have some arguments here. So once we upload this matrix, the app will automatically calculate that how much RAM do we have in our computer, how many cores do we have, and based on the memory that this sparse matrix is taking, it will suggest that how many cores at max should we use that our system doesn't crash. So if we provide all, it will do the maximum number of cores, and I can also provide whatever I want. And similar to the last step, we also have the location where we want to save it. So I will again choose desktop, and we have these other options as optional, that the expected hit size. So expected hit size is basically if someone has their prior knowledge or an idea about how many hits they are expecting. like depending on the diversity panel or depending on the traits, the expected hit size might change. As of now, we have kept 3 options, like approximately 10, 20, or 50. We have named small, medium, or large. So if some researcher has their idea that I want only this many, they can train their object for only those many hits. But all are recommended. Why? Because we are doing this training one time where we train for all the hit sizes. We will see in the next step, when we analyze multiple traits, for multiple traits, we might want that to use different expected hit sizes. So if we have this object trained once and for all, we are done, and we can use this trained object for any trait we have, even if for different traits we want different hit sizes. And this MAF threshold is basically the minor allele frequency. So inside our pipeline at one particular step, what we do is we clean the data a little bit. At one particular step, before doing that, we drop some SNPs, the columns of the SNP matrix, which has minor allele frequency less than 5%. This is also open to the user. They can change it like 10%, 20%, of course. Should be ideally less than 50%, because more than 50%, if it goes, it no longer remains a minor allele, it becomes a major allele. And this R square, this is basically an idea of how much variation of the phenotypic trait might be explained by the SNPs or explained by genetics. So if we have a prior idea, we might want to mention that. By default, it takes 0.5. So these parameters are all like fine-tuning our study if someone wants to do it. Otherwise, there are default options as well. Now, I have chosen all these parameters. I have chosen my location where to save. Then we want to give, since this is a time-taking job, depending on the size of this matrix, we want to give the user an idea how much time it might take so they can accordingly plan. So if we click on this calculate training time, it shows me that it takes approximately 22 minutes on my 12 cores and on this device to do the training phase. So if I click on train.gwas, the training will start and it will take 22 minutes to train the object. But for now, I will not do that. I don't want to wait 20 minutes and make all of you wait 20 minutes. I'll just turn off this session. And
one question, Devarshi, did you show them that the space, no, tab doesn't work for the...
Yes, I did.
You should choose the other one, which is...
Yes, I showed the error and...
Okay.
Yeah.
Sounds good.
And I also told that in the next version of our app, we will do the auto detect thing. Yeah. So once the training is done, what ideally should happen is it will automatically create whatever directory you choose. Inside that, it will automatically create this folder, which is named SVENETICS folder. If you open that, you will be able to see this SVENETICS_trained, this object. But for the sake of time, I have already given that object SVENETICS_trained in the box folder, so that in the next step, we can directly use that. So what ideally would happen is you press that button, it will take those 22 minutes of time and it will stop, and you will be able to see that this SVENETICS folder, and inside that, you will be able to see this object, which we will need in our second step. So to launch go to our second step, I'll just launch the app again. And as our regular practice, open in browser. It's taking a little bit time. I don't know why this should not. Oh, I did not exit the, sorry. So I'll launch this, open in browser. Okay, as of now, let's just do the regular app. So we talked all about the training part, and we have already saved the object we need. Now we want to run the actual GWAS. So that's in a different tab called Run GWAS, right? So here we have to upload two objects. One is this SVENETICS trend we just saved, and it is given in your box folder, this SVENETICS trend object, which should be created by the earlier step. Something's wrong with my computer. Let me clear the environment. Sorry for that, y'all. So let me see. I launched this. Yeah, now it's okay. Sorry for the delay, please. And so we browse it and upload this SVENETICS, our trained object, which once we have got it trained, we can use it to analyze as many traits as we want. And this trait file, we have a trait file. Since we are talking about multiple traits as a demonstration, we have two traits. So we'll upload this. And now you can see the point I was trying to make earlier, that for trait 1 and trait 2, we might have different expected hit sizes. And if we have that object trained for all the hit sizes, we can play around with this. Like for each different trait, we can mention our different expected hit sizes. The app will automatically catch that from the trained object if the trained object is trained appropriately and do the analysis. And again, to save, I prefer my desktop as of now. And it is already showing that this will also be stored in the SVENETICS folder, that one. So if I just click on run.gwas, so it's running. Shouldn't take long. Oh yeah, the GWAS is complete, and it again shows us the path where it is exactly saved. So we go back and we can see in SVENETICS folder, we have these different CSV files. So for each trait, we will create a separate output file, because I have also analyzed datasets with 50 traits. So creating one CSV file doesn't look very good. So we will create different CSV files. I think that provides more flexibility. And suppose you don't have this SVENETICS folder in your desktop. Suppose you have trained earlier months ago, now you have moved that to somewhere else. If you just provide desktop and run the GWAS, I just want to show you that even if you don't have the existing folder, so in your location, in my case, which is desktop, it will create this SVENETICS folder and then store the results inside that, similar to that of the trained object. So are there any questions till now? If not, then only I'll proceed to the how to interpret the outputs of the.
Yes. Do you hear me?
Yeah, I can hear you.
Okay, thank you. I just late to join the meeting.
Okay.
And I did not hear the session from the starting. So what I have not know is I don't understand how we can convert the genotype data to this suitable format for this applications or program.
Okay, so I think since you
excuse me, I just let you back to really sorry for that.
Since I think so it's probably that you didn't get to listen that we have a different app for doing that.
Yes, actually, I hear that you just change it through browser. I don't understand that browser. It whether it come from this app or from other. I don't understand that.
Okay, so basically we have a completely separate app for that. So the latest app I was talking about, this line of code launches that app that does all the training and running these GWAS. Is that clear?
Yes, actually, I understand that.
Okay. And to convert your numeric matrix to a sparse
Yes, I
matrix,
have.
we have a different app where if you upload your numeric matrix, it will give you back the sparse matrix. If you want, after completing this, which should be within 5 to 10 minutes, I can again get back to that app.
Could you demonstrate the dense to sparse converter app once more?
Yeah, I can. Okay. So this is the line of code that launches the dense to sparse converter app. So we just go ahead and here we'll naturally browse, and I will make this so that it shows me all the files. So this .mat file is our numeric format.
Numeric matrix, which content, each allele has been converted into 0, 1, 2.
Yes. So that's our numeric matrix. We upload it.
Could it be in CSV format?
I'm not sure if we included that.
Yes, it could be. See, is there a separate for the separator? Is there a comma option?
Yes, there is a comma option.
So it should.
Okay.
Yeah,
It should.
you can.
Thank you.
And
I have already said.
Right. For different files, the delimiters might be different. So try out one by one. If it's a mismatch, it will show that no snip columns found in there.
Yeah, I can show that once more. So basically, if I just go ahead and click this convert to sparse, you can see that it throws an error. So basically the numeric format
tries to, go ahead.
Oh, so yeah, it throws an error because the format we have right now, the file is space delimited and not tab delimited. So it cannot recognize the snips columns in the header. So I will just change it to space because I know.
How about you change it to comma? See if the same error appears.
If we change it to comma and click it, it will throw the same error.
Same error because it's expecting comma to separate numbers, but there's numbers are
same
actually
will
separated.
happen if I go to semicolon, it will throw the same error. So if I go to space and click this convert to sparse, it will do it in a second. And just reiterating, in the next version of our app, we will have the auto detect thing. So it can automatically detect what the separator is. So is the usage, did you understand it better this time?
Yes.
Okay, thank you. Okay, now I'll go back to the outputs we had after our GWAS. So the output files for each of the traits were stored in the CSV files inside the SVENETICS folder. Let us open one of these files. Okay, so this is how our output looks like. We have the first column as the SNP IDs. And our second column as MIP, this basically means the marginal inclusion probability. I will tell you how to interpret this. So marginal inclusion probability, you can think of as an importance measure of the SNPs. And it is a number lying between 0 and 1, since it's a probability. And higher the MIP, the more important the SNP or the hit is. So what output we provide is we provide the MIP for literally every SNP, and it's stored in a descending order. So the more important SNPs are here at the top of the file so that it's easy to see. And as we go down, we see that they gradually approach 0. And after some point of time, they will essentially become very, very close to 0 and absolute zeros. Like 10 to the power minus 25 numerically, that is basically 0. So now, how we interpret this is the higher the MIP, higher the more important the SNP is or the hit is. And there are the cutoff which we might want to decide is we have not made it very hard and fast as of now. Like if you have used different traditional statistical methods like FarmCP or Blink, if some particular p-value or q-value of some particular SNP is less than 0.05, we consider them as significant. In this case, as a thumb rule, we might say that, which is followed in several literatures as well, that if the MIP is greater than 0.5, we might consider that as a hit or as important SNP. So if we set that cutoff to 0.5, all the SNPs still here will be our hits. If someone wants that to consider that as 0.4, in that case, everything till here will be our hits. That's up to the user, but as of now, we can recommend a thumb rule that 0.5 is a pretty good point to start with. But yeah, not all traits will show the same amount of variability, and they might not have as high MIPs as, not all traits will show like the same type of high MIPs or low MIPs. Like if we just look at the other trait, you can see that it doesn't have those values as high as 0.99 or something like that. So if here we set our cutoff to be 0.5, we will just get these 3. So yeah, we try to keep that flexible, and that's not a very, very hard and fast rule. But yeah, 0.5 is a good point to start with. So that's how we interpret the outputs, the SNP IDs and the MIPs, just like SNP and p-values or q-values. We try to make it in a similar format in which people are habituated or where people are familiar with. So yeah, that was mostly what I wanted to demonstrate. And if you have any questions or regarding the usage of the app, please ask me.
One of the common issues that I think I see in the chat is that when you install, you get an older version of Bravo. So I've been checking, are generally hosts or the CRAN packages are hosted by a lot of mirrors.
Yes.
And it is possible that some of the mirrors are kind of old, they're not updated.
Yep.
So either you can use a mainline mirror, or is that code basically?
Oh yeah, you already posted in the chat. Yeah.
Yeah, about using, for example, the Iowa State also hosts a CRAN mirror.
Yeah, I had that as a backup. I kind of thought that this problem might come up. So this is the code you might want to use then.
I've been searching over the mirrors. I don't yet see anything holding still.
So should I paste it once more in the chat box so that?
Yeah, okay. Yeah, I can.
Okay, I pasted it once more in the chat box.
Oh, but maybe some of them can use it requires the source package. So
okay.
Better use this one, the
okay.
Iowa State one, because for Windows and
Okay,
Mac, it will automatically install the compiled packages, which would be faster.
Okay.
And also for, you cannot install, you may not be able to install from source on certain Windows machine if you don't have the developer kit installed.
Okay, I see. Yeah.
But the Iowa State mirror is up to date, and it has all the latest version as well as the compiled packages.
That's a new information to me as well. I'll keep it.
Devarshi.
Yep.
Yeah, may I ask a question?
Of course.
While you selecting the trait 1 for the small value, the 20%, and for the trait 2 is a 50%.
Mhm.
The selection of these limits affect the MIP values in the results.
It does.
So we have to check all three in all traits.
It's totally up to the user if they have any prior information that how many hits do they expect. Like we have seen in certain studies, researchers beforehand, they have an idea that this particular trait or this particular kind of plant like should produce at least these many traits or around these many traits, like different for soybean, different for maize. So if only the user has that idea, they should select that. Otherwise, we'll do the default one. Or yeah, in order to explore and find out, like get an idea that which one is good for someone's particular research, they might try all those and see the outputs that which are more favorable to the scientific narrative or something. It's always good to check all three, of course. But by default, we'll do the like medium one.
But if I don't have idea about the traits and about the significant SNPs, for an example, the yield traits, yield of wheat or rice is a complex trait. So it is the ultimate target. So if we go this trait, so we have to try all these three limits to check the important SNPs.
For
The
traits
number one,
complex, yeah.
but the second one, if we go the single trait, for an example, the number of spikelets, the number of kernels per panicle. So we can in this trait we can go for the medium values to check the SNPs.
Yeah, of course. If you have a complex trait, you definitely would want to try the highest one.
Okay.
Transcriptomic data. Okay. It can actually, yes, you can also use transcriptomic data, but not in this app. This app does not allow. It only allows either SNP or transcriptomic data, but not both. But the Bravo package does have a function where you can include both SNPs as well as transcriptomic data. Do you remember the name of the function? I think it's SVEN, right?
It's SVEN. It's included in SVEN only. Yeah.
Yeah. It's SVEN. So there is a function in the package called SVEN, and there you can pass a list of matrices, but that will require a bit of coding, and
Shumagda, I think you can post your last year slides. The details there.
Right.
You can just give the your website URL because it's clearly explained in detail. And
right. But we may want to take a step back because there the parameters are not tuned.
Yeah, that's
the way we have tuned
so
in this case.
So we can do, but the parameters are not tuned. So we have to do that manually before.
Yeah.
Yeah, but that's actually a nice feedback. We might want to implement this in the app in our next
version.
Next
workshop.
That's actually very good feedback. Thank you for that.
There's another question about, there are two questions. So one is if we need to convert the data into linear format, similar to the marker data. No, not for the transcriptomic data, because transcriptomic data are typically not sparse. They're continuous values. The SNP data are typically sparse because you may have only a few choices, right? 0, 1, and 2, and generally for a major allele, the major allele will be more frequent, so you can store that as 0. That's what we actually do. For gene expression data or transcriptomic data, those are continuous, so we don't need or we shouldn't convert them into sparse format. Typically, the gene data is much lower dimension than SNP data. You can have millions of SNPs, but what we have seen genetics data are, number of genes typically are in the thousands or tens of thousands. Before.
And the tool, the SVEN tool, actually allows nonlinear relationship for the gene expression data as well.
That your trait can increase or decrease with the gene expression, but it can do so in a nonlinear fashion. It can go up and down or go plateau, all those sort of relationships are possible. There's a question whether this method considers the population structure into account. It doesn't. SVEN is a multi-locus method, so it considers all traits at the same time. All SNPs at the same time. So it does not need to account for the population structure. In some other methods, typically one SNP or a group of SNPs are tested at a time. So the population structure need to be accounted for in Blink and PharmCPU, and that's typically done using the principal components, but not for SVEN. For SVEN, we do not need to account for. We do not need to use the principal components to account for the population structure. How do it compare? Sorry, go ahead.
Let me ask one question.
Sure.
Can we convert the GWAS format to the mat or this semantic sparse matrix format?
I believe so, because in GAPIT you need GAPIT gives you the numeric genotype data, right? Numeric format, right?
Yes.
And then you can
especially the genotype format.
Yeah. And then you can use that to in the dense to sparse converter. And please feel free to try this out while we have like another 50 minutes to go. And please feel free to try out these with your data sets. And if you encounter a problem, then we'll also know what are the possible.
I know that the major challenge is getting the appropriate format for the program. Yeah, because the half map there, the GWAS format, the
half map won't work. Half map won't work. VCF won't work with us. So you need to convert the half map or the VCF into numerical format. Tassel can do that, for example. But I think GAPIT can also do that.
What is the special merit of this app than the Blink and the PharmCPU?
These are new different methods. The main difference between SVENETICS and Blink or PharmCPU is that SVENETICS is a Bayesian method, so it considers it doesn't test one SNP at a time, because when you test for one SNP at a time, you may have to adjust for multiplicity, you may have to account for the population structure. But in SVEN, it considers all the SNPs at the same time. So it does not have to account for the population structure, and it also does not have to account for multiple testing. That's the major biological difference. Another difference is that in the SVENETICS pipeline specifically, all these methods have tuning parameters. In Blink, in PharmCPU, you have to give the window size, right? That's one of the major tuning parameters. And you have to have a good idea about the window size. Like different window size may give you different answers. In SVENETICS, what we have done is it also has some tuning parameters that are in some way related to the window size, like how fast your LD, how fast the LD decays, and also like whether the trait is complex or simple, some basic idea about that. And in SVENETICS pipeline, what we do is we train using the panel, we train the hyperparameters. So you don't have to manually specify the hyperparameters. It is automatically tuned on this diversity, on the particular diversity panel. Does that answer your question?
Yeah, but all the three are multi-locus GWAS.
They are
Blink
multilocus,
for
but in yes, they're multilocus, but in different sense. Blink still doesn't, although it is a multilocus, I don't think it tests like when it tests, it looks at a window. So it tests one marker accounting for the window, but it still tests, assigns one p-value for one marker. In SVEN, it's more complicated. If you have 1.5 million SNPs, then it considers a space of 2 to the power 1.5 million possible models, possible combinations, and tries to find out the combination of the SNPs that explains the phenotypic variations the best.
Okay, thank you.
Explain again the cutoff, how to decide on the cutoff for the mean. That's a very good question as well. Our default rule is 50%. So if you see a marginal inclusion probability of more than 50%, then that you can call it a significant hit, or you can call it an important hit. What is it? The diagram, the true false discovery versus true positive rate diagram that Deboshi showed, it was based on 0.5 as a cutoff for MIP, right?
Yes.
Yeah. So based on our studies, we think 0.5 works pretty well. But as usual with a hard threshold, the problem is, like with the p-value. What do you call a p-value threshold? Why should 5% be a good threshold? Why not 1%? Why not 10%? Similarly, if you want to use a lower threshold, 40% for the MIP, or if you want to use a higher threshold, like 60% for the MIP, that's also okay. What could be the comparison with other multi-trait, multilocus GWAS? So that kind of brings to the Blink paper. In the Blink paper, they compare their method, Blink, PharmCPU, to others, multilocus methods, FastML, and others. And they already showed that Blink and PharmCPU were performing better than those other methods. And hence we built upon that, and we just compared SVENETICS to PharmCPU and Blink.
Yeah, and we also used the exactly similar simulation setup as they did.
Blink, yes. So we expect that SVENETICS will be better than other methods as well.
Are you improving still now? No. Improving.
That
still?
because we are trying to make it more user friendly. So for example, we found that the delimiter, whether the numbers are separated by tabs or space or comma, we are going to next iteration, we are going to make that automatic. Like you don't have to specify what delimiter it has. The code will detect it. And also that's kind of, features improvement, but we are also going to include newer, more advanced methods. So for example, right now we are using this method works for traits, like you have the blues and stuff like that. Blues or blobs. But there are other traits that we are working on where there's no blue. It's a function. For each genotype, you have a function of response. And we are going to include GWAS for those sort of traits, where the response is not a single number, but a function, a curve, for example. Like your identify, you will want to identify genetic basis of growth curves. Different genotypes may have different growth curves, and you may want to identify SNPs that lead to those different types of growth curves, not necessarily just average growth or something like that. There's another question. I don't think it's a stupid question. I think it's a great question. The question is whether we need to apply a Bonferroni or false discover FDR type of adjustment to the marginal inclusion probabilities, and the answer is no. It's because, again, it's because SVEN or SVENETICS is it's like a, it's multi-locus, like it, not just multi-locus, it's like a whole locus. So it considers the entire genome and then tries to find out the best combination of SNPs. And in that process, it does already penalize the too many hits or too few, basically. So it's already penalized. It's already adjusted in that sense. So we do not need to adjust it any further, adjust the marginal inclusion probabilities any further. We do not need to do that. There was a great question on polyploid, whether the method will work on polyploid on species. The answer is not directly, but it can be. I mean, for polyploid species, for example, if you have hexaploid species, then you need to convert each marker into 5 dummy variables, something like, where each dummy variable will indicate which then basically count number of different samples, I think. So the short answer is the app won't work, but with some coding, the basis, a SVEN pipeline, SVEN function will work. But yeah, those are, we don't yet have a good method to tune the hyperparameters for those problems. So you need to kind of come up with your own tuning parameters, or wait for some time that we add the tuning feature for polyploid and gene expression.
There is another question. I think it's about whether allowing environment, the interaction with environment effect, I think.
No, not just interaction. I think it's also like whether we are using those information, right? So that should go to a pre-processing step. Like generally, a mixed effects model would be used to compute the best linear unbiased estimators or best linear unbiased predictors of genotype. And those blues or blobs should be used in this GWAS pipeline.
As the response.
As the response, yes.
Yeah, as the trait we are using.
As a trait, yes.
typically, yes. Spatial adjustment in terms for field data, you may also want to adjust for spatial trends, but those can happen before the previous step when you compute the blobs or blues. Another point to note is, I think it's if you have a good hardware, like typically when we run these methods on large data sets, on large panels, or panels with lots of markers, we use like servers or workstation with more RAM and more CPUs. You can do that as well. If you use a workstation or server, you can install Bravo there, and you can use if you have access to RStudio through server, or you can also launch our command line in the server.
Can I say something?
Yeah, go ahead.
Yeah. So yeah, for this app, we have tried to make as accessible as possible. So since it's just 2 lines of code, just base R is also okay. So RStudio, like even if you don't have it on the server, you can even run it from the terminal. It will launch the app. And also, the point Shomak just said, I want to mention that how huge effect the number of cores has. I just want to show you. So if I go and launch this app, Shomak, is my screen visible?
I just lost.
Yes.
Okay.
Yes.
Okay. So this training part is the heavier part. So if I go give this parse matrix and say I specify the number of cores to be, say, 3. And I will just calculate runtime to show you how it scales. So calculate runtime, it will take 94 minutes. If I make it double, so again recalculate runtime, it's approximately like not exactly 45 or 47, but almost half. If I make it 12, so basically again half of that, because we run the simulation kind of in a parallel way. So if you just double your number of cores, the training time will reduce to approximately half of that. So that's a huge advantage of having multiple cores in our part, because the way we compute, as many cores we have, it directly scales the computation time. Like we double the number of cores and our computation time becomes half. That direct scaling happens. So it's a scalable methods in terms of as many number of cores we have.
Also, a warning on that note is you also need good amount of RAM to
Yes.
scale them, because if you try to parallelize over
Hmm.
12 cores, but you only have 16 gigabytes of RAM in your computer, then it will crash.
Yeah.
Because,
And that's why we have that initial warning message. It will show you that.
excuse me?
Probably that was accidental.
Yeah.
So that's why we have that initial warning message that once you specify your number of cores, or once you just upload the matrix, it will show you that how much space it is occupying, how much RAM do you have, and what is the maximum safe number of cores to use so that the system doesn't crash. That's why we have that. So yeah, these are some options the user can always play around with. If someone has huge number of cores, huge RAM, they will do it in like an hour. Maybe very big data sets within an hour is possible. Or maybe overnight at least.
Yeah, so the expected runtime should tell you whether you should go for a coffee and come back, or whether you should just go home for the day and come back next morning to have results.
Yeah.
There's a question on chat which says asks whether genotype names have any restrictions. The answer is no, right?
No, we don't have any restrictions on the format. Yeah.
They can have space, character, or they can also have parentheses.
Parentheses, we take care of that, yeah.
But just sometimes what we have seen is that the genotype names in the trait file, oftentimes they do not match with the genotype names in the SNP file, which is a problem, if we don't know how to match them, then those genotypes would be dropped.
Yeah. Since we are automating it, so one of the typical things we have seen is in the trait file, we have a hyphen, and in the SNP file, we have an underscore. So if a human is doing, they will understand that it's the same thing, but in an automated pipeline, they will not recognize it and just drop that.
And also sometimes they are different, like a hyphen
Yes,
versus an underscore.
yeah.
Sometimes they are different. So it's not clear. Oftentimes it's not clear whether slight mismatch, whether they're the same genotypes or whether they're actually different genotypes. We go with a safe option that maybe they're different, just drop them. How do we get Manhattan plots? Yeah.
As of now, we don't have that in the app, but it's not very difficult to generate, but hopefully in the next iteration of the app, we'll have the Manhattan plot. Like, of course, we don't generate using p-values. We use MIPs to do that. And once the paper is there, we have some kind of Manhattan plots in the paper as well. And definitely, that's also a very good point. Thank you. And we'll try to incorporate in the next version of the app, which will also, along with the Excel files, like download a Manhattan plot in the directory.
And probably we shouldn't call it a Manhattan plot.
Yeah.
It will be very sparse, so we should maybe call it
It
plot. Maybe.
Very discrete, something else. Yeah, I faced these questions in one of the poster sessions as well, that how to get. So we can get.
Yeah. We will add this feature one by one. So yeah. And I think you are all subscribed to the mailing list. We will email any updates or package version updates to the mailing list. But we promise not to send you too many emails. Just when very sparingly when packages are updated, or maybe the paper is published. That's all.
Yeah.
And Eddie, will there be a feedback form after this workshop?
No, but I can adding up. We can send later on. We have the people register. We can send that.
Sounds good. Thank you. So yeah, we'll send you a feedback form, and please, it's optional, of course, but we'll be very glad if you. Not bad. I'm saying. What's the word I'm looking for? Will be. Grateful. Thank you. We'll be grateful if you could provide the feedback after for this workshop. Thanks.
Yes, definitely. Like, it will help us do as many changes in the next update as possible. I have already noted down the verbal feedbacks I already got, but yeah, it will be great to have written feedback as well.
And also, I think if you try to use SVENETICS for your own data sets and you encounter problems, please feel free to reach out. Feel free to reach out to any one of us. We'll be very happy to sit with you and debug.
Yes. Our email IDs should be there in the R package? Or?
Yeah. Our package.
Oh, no, your email ID should be there. Or no?
No, I think everybody's email ID. But also the poster for the workshop, it probably includes your email IDs.
Yeah. And the slide, of course, includes mine and Srijit's email ID. So it's okay if you reach out to any of us. Yeah,
and you can Google any of us.
Yes,
we are all there on the iWAS
it should be pretty easy to find. Since R is open, I replying to the last question that if you plan to make the underlying R code available to the public, so R is open source software. So on CRAN, like the codes are available. Like an easy way to access it is to navigate to the GitHub repo. All the codes are there. All the codes are already there.
Let me see if we have the GitHub repo link.
Oh,
I pasted it some times ago here, but yes, and if you're trying to make pipelines or trying to create your own pipelines, and you would, if you have any questions on how to integrate it, we'll be happy to sit with you and just show you maybe how to integrate SVENETICS or SVEN in your pipeline.
yeah, of course, you're welcome. You're welcome. And thanks for the excellent feedbacks as well.
Okay, I think, does anybody else have any other questions? Or maybe we can call it a day. Yeah, I think that's pretty good. Yeah, that sounds good to me. And please feel free to reach out to us with any questions you may have or any help you may need with analyzing the data or using SVENETICS. We'll be happy to help. All right, so thank you very much again for attending this workshop, and please consider providing feedback later on. And yeah, that's all. Thank you very much. You have a nice rest of the day.
Thank you.